Yeah.
Hello, everybody. Good morning, good evening. Welcome to BioacousTalks, our first session of spring 2025. And I was trying to think of a good metaphor, but Nadège Aoki, who is joining us here from Woods Hole, will be talking to us about the alluring effects of coral reef soundscapes, just like we were all drawn to the presentation that she's about to give today. So here we have all settled in this Zoom room to learn about coral reef soundscapes and how healthy soundscapes matter for coral reef conservation. So really excited for your presentation, Nadège. But before that, I just wanted to zoom out and just say, for those who are BioacousTalks regulars, I see many of you in the chat. Welcome back. And for those of you who are new, super excited to have you join us here. This is a bi-weekly presentation community session. We're going to be meeting every 2 weeks now through July for different presentations, meeting different researchers in the field to learn about exciting new studies, new tools, and basically different things that will inspire us all in the field of bioacoustics. So we are, really pleased and thrilled that you were able to join us, Nadège, to share what you've been working on these last 5 years as part of your PhD at Woods Hole. So I'll now hand you the floor. And for anyone who has questions for Nadège or for any curiosities that are sparked during her presentation, please use the Q&A, which you can see at the bottom of your Zoom screen. And we'll have time at the end of the presentation for questions. So with that, floor is yours, Nadège.
Awesome. Thanks, Ben. You guys can hear me. You can see my slides, all that. Awesome. Great. Well, I'm really excited to be here, and it's really nice to get to talk to a, acoustics focused audience. It's not always the case at conferences and things like that. So I'm excited to talk to you about coral reefs, their soundscapes, and my dissertation research. On that note, I thought I'd start with just a quick kind of intro to who I am and sort of what got me involved in acoustics research in the first place. It's really cool for me to be participating in the BioacousTalks from the Cornell Lab of Ornithology because I'm a Cornell alumna. I did my undergrad there, not doing anything acoustics related at all, but I did work at the Museum of Vertebrates, which is housed in the Lab of Ornithology. And I was working with these very cool weakly electric fish that generate electric discharges and can sense electric fields. And really cool thing about them is those electric discharges are species specific, and you can use them to diagnose otherwise cryptic species. So that work really got me interested in the sensory biology, sensory ecology of aquatic life. I've also spent some time working at the Smithsonian, at the Natural History Museum in DC, studying deep sea worms, very cool. Have spent some time at the National Science Foundation working more on sort of the science policy and communication side. And for the last 5 years, I've been based out of Woods Hole, Massachusetts, working in the MIT Woods Hole joint doctoral program advised by Dr. Erin Mooney. And now my work is really focused on the soundscape ecology of coral reefs. And I kind of said that all to also just plug that I am probably finishing my dissertation at the end of this calendar year. And I'm currently really open to and interested in looking for opportunities for future projects and postdoctoral work outside of this. So if you like anything I say in this talk, if you've got a cool idea around acoustics or reef science and you want to chat about it, please reach out to me. Okay, got my little plug out of the way. So as probably a lot of us on this call are aware, or maybe not, Coral reefs represent the most diverse marine habitats. So reefs support close to 25% of all marine life. And they also support billions of dollars worth of critical services for human communities. So reefs are really important fisheries for coastal communities. They provide protection from storm surges and other damaging natural disaster events. And they also obviously are really beautiful and important sites of cultural significance and bring in a lot of tourism money to a lot of coastal economies. So reefs are very important and valuable. However, they are also in crisis. So as climate change accelerates global reef degradation, we are seeing increasing frequency and intensity of stressors on reefs. And those can come in the form of marine heat waves that can drive coral bleaching and death. We see increased spread of coral disease outbreaks and other things like ocean acidification, pollution. There's a lot of stuff that is harming reefs right now. And that makes the need for us as researchers and stakeholders to better understand the dynamics of these vital ecosystems and at the same time try to develop actual practical and scalable interventions to help reefs stay resilient and rebuild after disturbance. It's all a very urgent need right now. So today I'm going to be talking about coral reef soundscape. So I'm going to be really thinking about how what we can hear and listen and record on a reef can tell us about the ecosystem's health and the function of the ecosystem. And also think about, okay, if we know that these sounds are really important to these ecosystems, is there a way that we can actively harness them for restoration or conservation purpose? So I'm kind of dividing my talk a little bit into sort of two sections. At the beginning, I'm going to talk a little more broadly about reef soundscapes, their characteristics, and some of the knowledge gaps that we see there when we're trying to do effective ecological work in the coral reef soundscape space. And then I'm going to pivot and talk a lot about coral larvae and sound, which is the topic of my dissertation, and a couple publications that I'm going to sort of walk through. So just to get us started, I just want to orient to what do coral reef soundscapes sound like? So what are their general characteristics? I know we have people here who are coming from the terrestrial space and just very different aspects of acoustic work. Coral reefs, while they vary a lot, obviously in space and time, there are general characteristics of them in that reefs are typically very acoustically rich habitats. So there are a lot of animals on reefs that are soniferous and produce sound. And typically what you see on a reef soundscape is domination in the higher frequencies. So between like 2 to 20 kilohertz or so, you get a lot of noise from invertebrates such as snapping shrimp. So there are lots of species of shrimp like this one here that have a really big claw that when they snap, it produces these very high amplitude, short duration, broadband snaps. And you can see those in the background of the spectrogram here as these like tall, thin lines. And then we also have all sorts of fish on the reef that actually vocalize and produce sounds either actively by strumming on muscles on their swim bladder, or sometimes, as byproducts of activities like feeding, you'll hear sounds from parrotfish scraping on the reef or things like that. So I'm going to play this 5-second clip that hopefully you can all hear, and you should be able to hear the snapping shrimp as sort of crackling in the background, and then the fish sounds as kind of a strumming. I'll do it one more time because that was a very short clip. Right, so kind of cool. And I mean, it was really wild to me, honestly, just learning that fish make those sounds in the first place. I think a lot of people don't even know that. And really what we've kind of started to tease out from the last, maybe 15, 20 years or so of passive acoustic monitoring on reefs as that acoustic technology has become more accessible and available, is that there is a fairly consistent relationship between sound levels on a reef and other metrics of reef health or the ecosystem quality. So one of those metrics that's very common is the percentage of the benthos that is covered by hard coral, so living coral. So when that's higher, obviously you've got a more like healthy reef that's covered in coral and has a lot of structure, and potentially then it has a diverse, thriving community. And so we see like here, a typical relationship like this where as overall sound levels increase, you get associations with more coral cover, more fish abundance and diversity and things like that. And the thing is, obviously, this is cool that we see this and that we can then, have some idea that monitoring the soundscape does reflect something about the ecosystem. But just looking at, the broadband sound levels doesn't necessarily actually tell us anything about what kinds of biological activity are happening there. And obviously, there's lots of other stuff in the soundscape, like, wind and weather on the water, and even negative inputs like anthropogenic noise from boats that can stress out animals on the reef. And there's a lot of all things that can elevate the local sound levels without necessarily corresponding to positive kind of interactions or activity on the reef. So when you start to decompose the actual contributors to the soundscape and start looking at the biological sources, that's where you start to get more information about, okay, like what actually differentiates healthy reef ecosystems from more degraded or less healthy ones, and where do you see that in the soundscape? So this is a cool study from Tim Lamont, I think Tim Gordon, now that he got married, looking at reef sites in Indonesia and basically taking recordings, and they identified these 10 different, stereotype sounds, then gave them these cool fun names like knock and grunt and whoop. And they looked and audited through a bunch of sound data recorded from reefs that they categorized as sort of healthy versus degraded, and also reefs that had been restored through an active coral outplanting effort. And what you see there is that the actual faunic richness and the presence or absence of these different fish calling types does give an indication of what the category of the reef is. And a really cool thing is that the healthy and the restored reefs both have, sort of similar characteristics in terms of the fish calls that you see there, and the degraded ones, right, they have different ones. So the degraded is the orange, and the purple and green are the healthy and the restored. And so that tells us that, okay, that it's not just like there's something going on in the soundscape here that's really reflective of like restoration and successional processes that are happening, and when you can detect recovery in the soundscape, which is really cool. But you'll notice, right, that those sounds were not assigned to any particular species. And that is because a lot of the sounds that we record on reefs are still unknown and unclassified. So there's maybe only, 100 or so, I think, reef fishes and or other species that have had their sounds actually recorded and, matched to the species that's producing them. And obviously that makes it very challenging for us to look at a soundscape and say that, any particular species is contributing to these cool characteristics and acoustic diversity, because we just don't know what is making what sounds. And so and also, it is very hard to go through hours and hours and hours of data and try to pick out individual fish calls in the first place. So a way that our lab at Woods Hole is trying to address this is, first of all, we made a big push in the last few years to do a big labeling effort of tons and tons of fish calls. So this meant having undergrad students, graduate students go through, listen to audio and look at the spectrograms and create these labels that you see here, where the fish sounds are boxed in frequency and time, and we know what that, audio clip sounds like. And so this has resulted in a data set of, I think, like 55,000 labeled calls that have been curated for this data set. This is all data that's coming from the US Virgin Islands, where we've had like a long-term continuous soundscape monitoring project going for pretty much the last 10 years. So it's an amazing data set, but obviously it's like really, it's a huge amount of data to try to comb through. And you can see from just from this, 10-second clip here that has like, I don't know, 15 or so calls in it, that the actual calling rates we see at some of our more active reefs are really, really high. And so this data set has allowed us to produce some work. So these are several publications. I didn't lead any of them, but I did help out with some, thinking about how we can match up now that we have some ground truth actual calling rate data, and see how that matches up with other metrics like acoustic indices and sound pressure levels that have been previously sort of used as proxies for biological activity. Now we have some real data to look at when the fish are calling, and how those trends change each day, and with the lunar cycle and things like that. And then the next step in this is can we use this training data set that we have and build, automatic detectors for reef fish calls. And obviously the next step after that would be if we then can have audio and visual confirmation of which fish is making which call, can we build a classifier and, do all of that automatically? Obviously, we're not there yet, but I just wanted to kind of, again, shout out work that I did not lead, but have been tangential to in my PhD. This paper came out in the Journal of the Acoustical Society of America in JASA last month, led by Seth McCammon, who's an assistant scientist at Woods Hole. And in this paper, they've use this massive training data set of fish calls that I mentioned and trained a YOLO-based convolutional neural network to detect fish calls across data. In this graph here, you can see that the upper graph shows basically performance of the detector in counting fish calls across 24-hour periods at 5 different reef sites, and the bottom graph shows comparison across those same sites, but just looking at sound pressure levels in the recordings. And you can see that we get, very similar diel trends here, where we have characteristic peaks of chorusing on the reef at dawn and dusk, and those come out. But there's also, clearly some differences here in terms of where we see higher sound levels is not always where we actually see activity, and the differences between the reefs are also, a little bit like they don't exactly match up between the two. So it's really interesting as we start getting into these other metrics and ways of analyzing our data sets for biological activity. Machine learning has a lot to give us there and allows us to start pinpointing, in more targeted ways, areas where the fish are like actually contributing to the soundscape. So that was so that was my, pivot at the little detour at the start to just talk about soundscape ecology on reefs in the general way. Now I'm going to zoom in very specifically to, all right, we have a real understanding now that soundscapes on coral reefs are both reflections of the community dynamics. So you have sound produced as a byproduct of a lot of activities and interactions, but sound is also an influence and a driver on those community dynamics and interactions. Even animals that don't produce sound are often sensitive to it, and that sound is an environmental cue and factor that will, drive things that they do. And so we know that sound plays a really vital role in driving a lot of ecologically important behaviors, whether that's, mating behaviors, territorial defense and competition between animals. We have animals that produce sounds in order to deter predators. And obviously the sounds of a reef are a really kind of, they're beacons underwater that allow animals to navigate towards them. And where this really comes into play is when we think about larval dispersal and settlement. And a larval dispersal is such a really important process in marine communities because so many marine species go through a larval stage when they're juveniles, where they are either carried by currents or actively swim and seek out habitat that is suitable for them to settle in and make their home and grow out for the rest of their lives. And increasingly, we're seeing that sound is a really important cue during that stage. So just again, to kind of orient here for those who are not maybe familiar with coral biology or with benthic ecology of marine invertebrates, this image here shows the life cycle of a coral. Corals are animals, cnidarians related to jellyfish. So the way that a coral baby develops is adults produce gametes, and those gametes become fertilized embryos, and that can happen either outside of the coral or actually inside. We have these two categories of corals that we call either broadcast spawners or brooders. Not super important. Either way, you end up with what we call a planula, basically a larval form of the coral that is capable of swimming and will spend somewhere between hours, days, weeks in the water column as a planktonic form, and then experience this settlement process where they could drop down out of the water column, attach to the substrate, metamorphosize, and then go on with their development. And this field of settlement ecology is, really, there's so many factors that go into the decision-making process of a larva when it is choosing where to settle. There are tons and tons of really big questions that are still left to answer here. And that has a lot of implications for things like coral restoration efforts that are trying to make degraded reefs or new, artificial reef sites attractive to larvae. We have to think about what makes them want to settle, right? So this is what coral larvae look like. These are maybe just a couple days old larvae of the rice coral, Possipora damicornis. And you can see how active these guys are as swimmers, within just a little, not long after being released, they will start to exhibit these searching behaviors where they look for where they're going to live. And during this process, larvae are really reliant on sensory cues from their environment to trigger that substrate attachment and metamorphosis process. And these sensory cues are really important because without them, larvae can actually delay metamorphosis for days to weeks, or they may not settle at all. So you can have larvae that if you keep them in the lab in super clean water that doesn't have any reef chemicals or cues in it, and they will just not settle, not settle, not settle. And eventually, that's super risky because the longer that you as a larva stay in the water column, the more risk that you're going to be carried out to sea to somewhere that you don't want to be, or you might get eaten by a butterfly fish, or you might just run out of energy. So these corals, they have these lipid reserves that they come out with as larvae, and then they're limited. So they really need to settle soon. And delaying that process can potentially have negative effects for them later on. So this table shows what some of the documented coral responses are to some different sensory cue types. So coral larvae have been known for quite a while to really prefer to settle on surfaces that have previously been colonized by crustose coralline algae, or surfaces that have microbial biofilms on them. We don't know exactly, we're still, there's still a lot of questions here as to what metabolites or what biochemical compounds the corals are responding to, but this is already something that's been put into practice broadly for coral restoration practitioners, where if you are trying to get corals to settle on surfaces, you make sure that they're preconditioned in seawater with crustose coralline algae beforehand, or your corals will not settle at all. And then obviously light is also a factor here, so is pressure. And what I'm going to be talking about is the influence of sound on coral larval settlement. So there have been a couple of really neat studies that came out in the last 15 years that showed, first of all, that corals exhibit phonotaxis, or directional swimming towards a sound source. And that was really the first study that came out working Curacao that it indicated that coral larvae are sensitive to sound. And then Ashley Lillis, a former postdoc at Woods Hole, who I believe also gave a BioAcoustic talk a year or two ago, she found that when you put larvae on reefs that have sort of differing ambient levels of sound, that they settle preferentially, in the presence of those higher ambient sound levels, which is really, really cool. And this knowledge that, okay, natural soundscapes seem to have an influence on larval settlement rates. What about kind of artificially enriched soundscapes? So acoustic, the use of sound playback, or I like to call it acoustic enrichment. That term is sort of starting to catch on, I think, as a restoration tool, has been tested in other populations. I know in birds, there's been a lot of work on this idea of conspecific attraction or heterospecific attraction, where, if you play the sounds of a particular bird species, do other individuals of that species, are they attracted to it? And, could that have restoration impacts if you're trying to colonize a particular area with a particular bird population? And you can see from this chart here, which summarizes, some of these applied studies of animal responses to replayed sound, and what has been studied in different taxa. You can see there's been a lot of work on birds, a good amount of work on fish, and even some mammals and amphibians. But when we get to these other marine invertebrates, there's very limited data. Not a lot of people have looked into this, and really with corals, nobody had ever really tested that. Okay, if you play sound to them, is that sufficient to induce a settlement response? So this, has all led me to work that I've been doing in my PhD and examining whether coral larvae settle in response to recorded reef sounds if you play them back in situ at a degraded reef site. So for the rest of the talk, I'm going to run through the sort of experimental setup and results from these two studies that I co-authored in the last year. And hopefully there will be time at the end for questions. So first of all, when you're trying to do a study like this underwater, you run into the issue of how are we going to play these sounds underwater, because that's not a trivial thing to do. You can't just walk out and dump a speaker. You need to have a way to power it. You need to have a way to time your things, and that all has to happen in the ocean, right? So for the last few years, we've been working very closely with engineers at Woods Hole. That's a real strength of the institute here, is our connections with engineers to develop tools to deploy in the ocean. And we've developed what we call the Reef Acoustic Playback System, or the RAPS. We don't call it the Coral Reef Acoustic Playback System because that comes out to a different acronym that you can imagine for yourself. And this is a robust, customizable, solar-powered speaker system, primarily made with off-the-shelf components, with the idea that we could potentially hand this to a user, whether that's a restoration practitioner or a manager or anybody, or another scientist or researcher. You can use this and schedule different sound files to play when you want, and that it can run itself, ideally indefinitely. We've definitely had them working for weeks at a time right now. So in this first study that I used the RAPS for, we were looking at settlement responses of the Porites asteroides coral. So this is also known as the mustard hill coral. It's pretty common in the Caribbean. It's a little bit of a weedy coral, one that has been doing pretty well, relatively to some of our other more endangered species. It's also a brooding coral, which, like I mentioned, these are corals that do all of their fertilization and development of larvae internally. And so that means that they're as easy as a coral can be to collect larvae from. You basically just bring in your colonies, observe them during the time of the lunar cycle when you expect they'll spawn, and you collect your little guys, which look like this in the lower left-hand corner. After we had our larvae, we portioned them out into little settlement chambers. And these cups are what basically allowed us to isolate the influence of sound from other possible sensory inputs on the reef. So I put a standard number of larvae in each cup and added 0.2 micron filtered seawater. So removing grazers, removing other chemical and microbial influences from that reef water. and then added a standardized settlement surface to each of those cups. And obviously, while the cups don't transmit sound perfectly, because the acoustic impedance of the polypropylene was pretty close to that of seawater, we considered it, as good as we were going to get to allow those external sound cues to penetrate the cups and get to our larvae with minimal transmission loss. And once we had those settlement chambers prepped, basically our team put them on our different treatment reefs, and I'll go over our treatments in a moment. And then after 1 to 3 days of exposure, we collected them again and looked at whether or not the larvae had settled. And this on the right, this image shows what the corals look like when they settle. Very different, right, from those larvae that you saw earlier. They flatten out and they immediately begin putting down these septations that indicate where their skeleton is going to grow. And we did this twice in the summer of 2022, once in June, once in July. In this experiment, we had 3 reef sites we were working on the south side of St. John in the Virgin Islands. So 2 of these sites, Salt Pond and Cocoloba, are what we consider to be degraded, very low levels of coral there and low ambient sound levels as well. We also had a sort of ostensible positive control site, tektite, where the ambient sound levels are a lot higher. And essentially, we played back sound with the wraps at Salt Pond, and we did not play anything at the other 2 sites. Our sound playback treatment was a 12-hour overnight cycle, so that allowed us to play sound for from 6 p.m. to 6 a.m., and then we let the system recharge from the solar panels during the day. We've actually, since this study, it's kind of expanded the lifetime of the system, where you can run it for something more on the order of 18 or even pretty close to 24 hours of sound playing continuously without the batteries running out. But we were fairly conservative this first time around. And the sounds that we played back were recordings from the same reef area. So from our tektite reef, which is our really active, high quality reef site, but from 10 years ago. So that's what, having these long-term data sets allow us to go back in time and get these soundscapes that represent an older baseline for these reefs. And we randomized these, played a different treatment each night. These pictures just show you a little snapshot of the 3 sites we were working at. Not really important what these pie charts represent, except for that the white slices are how much hard coral is in the area when we surveyed it. And so you can see that Salt Pond and Cocoloba have very low rates, very low percentages of hard coral cover. Tektite has much higher, and you can even see some of those orbicellid coral heads kind of in the background there. So those are our 3 sites. And this image shows what the setup kind of looked like. So we had our speaker on the benthos, and then we placed cups of larvae at 4 different distances from the speaker, and we measured settlement at those different distances. And we used 4-channel hydrophone arrays to measure both pressure and particle motion at each distance. We used the gradient between each hydrophone to give us the particle velocity and acceleration. And getting a little more technical into the acoustics, right? But the particle motion, actual vibration of particles in a sound field is most likely to be the relevant sound stimulus for invertebrates and other animals that don't have a gas bladder or chamber that allows them to detect pressure. So it's really important in studies of marine invertebrate sensory ecology that you quantify particle motion in addition to pressure. So this video kind of shows what the setup looks like underwater. The big blue thing is our speaker, and that's me and my advisor, and you'll see us attaching our larval cups to rebar stakes. You'll also see in a minute the 4-channel hydrophone arrays that we put out at our different distances, and you can see this site is, it's covered in turf algae. This site, we just went to another clip, this is our healthier site. And yeah, you can see our little cups, our speaker. Okay, I think, is it? The cups float, so you had to make sure that you had the bags upside down when you were attaching them. So what do we find? Unsurprisingly, when you put a speaker underwater, playing reef sounds as loud as you possibly can, it has a pretty noticeable effect on the soundscape. So this is a 24-hour spectrogram of our playback site, and you can pretty obviously see when the speaker was on, when it wasn't. I'm actually going to play a quick like 10-second clip, and you'll hear like 5 seconds of just the ambient sound of the reef, and then the speaker comes on and you actually hear it start playing fish sounds right away. So, that's what we were hoping would maybe that these larvae would sense that stimulus and respond to it. We also, when we looked at the particle acceleration and how it decreased as you got farther away from the speaker, we see, what we expect, which is basically an exponential decrease with distance. But we did also see that our low frequency particle acceleration levels remained elevated at our even at our 30-meter distance from the speaker compared to either of our control sites. And when we looked at the spectral content of the 3 different sites, so this power spectral density shows the frequency content of just the unenriched soundscapes of each site. So the blue is Tektite, the red is Salt Pond, the gray is Cocoloba. You can see they're very similar. Tektite has a little bit more of a bump here, which we might expect because there's potentially elevated fish activity already happening at that site. And then when you turn the speaker on, it obviously significantly changes that spectral density. And you notice that the nighttime playback amplified our sound levels, but that power and change was not exactly equally distributed across the spectrum. And that, has a lot to do with the limitations of the speaker we're using and the transducer. It just wasn't great at doing reproducing frequencies below, 100 hertz or so. And that just speaks to the idea that in this kind of study, we're not trying to exactly replicate the natural soundscape. We're trying to do it as close as possible so that we elicit a response from our target organism, right? And it's just important to note that when you're doing a playback study, whether or not it's in the field or in a tank, just by virtue of recording it, amplifying it, then sending it back through a transducer, it's not going to be exactly the same as what you record from the natural environment. And what did we find with our settlement results? So in June, after 3 nights of exposure to these playback conditions, we did see significantly more settlement at our playback site, which is in red, than either of our control sites. In July, we saw a similar trend, although we did just missed out on significance. We didn't have, we're limited on larvae here. I didn't have as much replication as would be ideal. But either way, we did see highest settlement at our playback site compared to either of our controls. And this is just combined data from both months with the mean settlement rates here in this table. And you can see that they're about twice as much at our playback site compared to our controls. Yeah. And then when you looked at how settlement changed as you got farther away from the speaker, we generally see the trend that we would expect. And what we would expect to see, right, is that at our gray and blue here, which are our control sites, there's no actual sound source there. So distance isn't really meaningful. So you'd expect settlement to just be the same at all of those distances. At our playback site, we expected settlement to decrease as you got farther away from the sound source. And we did see that, except for at this 1 meter data point where we saw the opposite. Settlement went down. It was still elevated above controls, but not as high. And there's there's a couple of reasons this could be. One is that, we might not have enough replication here to really resolve those trends above natural variability. Also, it could be that at that 1 meter distance where we were blasting the sound as loud as possible, that there could be some sort of inhibitory effect actually at that close distance. But we really don't know enough about the mechanism of the larval sensitivity here to say anything definitive on that. But we did see that settlement it decreased with distance from the speaker and remained elevated above controls out to 30 meters. I was also interested in thinking about how the timing and the duration of exposure to a sound cue affected settlement responses. So while the majority of our cups were analyzed after 72 hours, I did pulse them at 24 and 48 hours and saw that sound generated higher settlement responses starting at 24 hours and continuing throughout that period. And this is important to just remember because we found some results later on with another species that showed the opposite trend. So in summary of this first study, we saw an average of 2 times more settlement on an acoustically enriched reef compared to controls. We saw that sound had persistent effects within 24 hours of exposure to the cue, and that our speaker system was able to enrich the soundscape noticeably in both the pressure and particle domains up to 30 meters away from the sound source. Okay, I'm just going to try to speed through this second little study that we followed this up with. Here, we were interested in looking at a second species. Obviously, doing this with one species does not necessarily mean it's generalizable to corals at large. For this one, we used Favia fragum, which is a smaller, again, another brooder, very similar larval collection setup for this one. And here our experimental setup was a little different. So we still did a field experiment, but we basically just had one field site and with one distance, 1 meter away from the speaker where we played sound, one site where we didn't. And then we also did this in a tank. So we put them in an aquaria setup and put a speaker in there to see what would happen. There's definitely relevance of, there's a lot of work on coral settlement that's happening in like land-based nurseries. And so figuring out whether or not this is an applicable tool to use in a tank where the acoustics get really funky was a priority. And here are settlement results. So on the left is the results from the aquarium, on the right is from the field. Gray is our sound treatment, white is our controls. And you see here that after 24 hours, we had settlement happening at higher rates in our sound treatments than we did in our controls. But then at our next time point that we checked after either 48 or 72 hours, that effect basically went away. So our impact of sound was only significant within the first 24 hours. And if you remember a couple slides back, that's completely different from what we saw with the Parietes asteroides species, where within 24 hours, they were settling more with sound, and that continued every time we checked on them. Here, it was only evident at our first time point. And so this makes us want to, okay, go back and think about what's the difference between these two species. And they actually have very different life histories. So this dispersal period, this really depends on kind of how well your larvae are provisioned, and it varies a lot based on different species. So the Favia species, the second one that we studied, are known to settle fairly quickly, so within like 36 hours of being released. And the asteroides species is not, they're not quite as well constrained in the literature, but we know that they're competent for very quickly after they're released, and that they can potentially spend much longer in their larval duration. And they're also just like, they're fatter larvae with more lipid reserves and potentially have longer to find somewhere to settle. So our, kind of hypothesis from this is that potentially the Favia are maybe the most sensitive to positive settlement cues during that first 36-hour or 24 to 36-hour window. And so they're probably the most discriminatory about where and when they settle during that time. And then outside of that window, they may start to get a little desperate and basically will settle anywhere or fail to settle at all. And that's why maybe we saw an effect of sound early on and then it went away. But obviously, this is something that really it prompts more questions, I think, than it answers. And that's, kind of the theme of my conclusion is that there's a lot of questions here to still answer. So in summary, from the second study, we found a second species that would settle in response to replay sounds, but the effects were variable with species, and you really had to take life history and the characteristics of the species you're working with into account. And a cool other thought is that, all right, like the tank acoustics, although they are distorted and not really comparable to field sound environments, it might be sufficient to induce settlement in a nursery setting. So it might you might be able to reproduce the soundscape well enough for the corals to respond. So yeah, awesome. I've, solved the coral reef crisis. Everybody can go home and we can just put a lot of speakers out and that'll save the coral reefs. Nope, that is not true. And we're definitely nowhere near being able to do that. Acoustic enrichment is not a silver bullet for the coral reef crisis. Even as we are rapidly trying to understand these ecosystems, they are experiencing these massive die-offs and stressors. So this is bleaching at Tektite Reef, our like lovely healthy reef that we have been monitoring for so long, and it saw massive bleaching in last year and or 2 years ago, and also even this past fall. So it's disheartening in some ways to know that this declines are happening so rapidly, but by studying the role of the sensory landscape and adding sound into that understanding in coral ecological processes, it can help us understand why and when reefs are failing and maybe identify possible intervention points. And here I'm going to put up a lot of text regarding questions that I still have that I think are really important to address. So there are still these fundamental questions of the biology and mechanism here of what are the frequencies and thresholds for optimal coral responses? How are the coral actually hearing? We have hypotheses about related to the cilia and hair cell structures that they have in their epidermis, but like we don't know. And then what are the community level effects when you put a speaker down and you try to enrich the acoustic environment? And then it's really important to know, like, is failure to settle really where the bottlenecks are happening on some of these struggling reefs? Or is it more post-settlement mortality that's preventing juveniles from recruiting to the adult population? And then, by addressing these fundamental questions, you can start then applying it to a restoration context and start thinking, okay, so which species or which reef sites would be good targets for an acoustic enrichment approach? And how can we structure our restoration approaches so that we are maximizing long-term survival of these settlers after they land? Um, and all of these, these questions are not linear. They feed back into each other. And really right now we have to be sort of trying out some of these restoration techniques and interventions while we're also doing the fundamental research at the same time, because we don't really have time to get everything sorted out before we try to implement some of these approaches to try to save reefs. And I don't have time. I was going to try to talk about some of the stuff we're doing in Hawaii. I just didn't really have time to fit it in, but we are continuing this work at Woods Hole. We're working on optimizing our technology. We're continuing to iterate it and trying to make it more user-friendly. We've been expanding our work and trying to look at more species that are endangered, or, more like targets for restoration, especially those broadcast spawners that are sort of the other category of corals. We're also building partnerships with the Nature Conservancy, with the NOAA National Marine Sanctuary System in Maui, and obviously thinking about how does sound work in the context of all of these other environmental forcings, like the chemistry of the water and the hydrodynamics locally. That is all really important when you're starting to actually think about this in an applied sense. So I want to acknowledge, my funders, as well as all of the tons of people who have helped me throughout my PhD. And I'm happy to take any questions. Thank you very much.
Thank you so much, Nadège. And from the flow of emojis, I could tell just how much everyone appreciated your presentation and really mesmerized by this amazing relationship between larvae and broadcasting reef sounds. So we have some questions in the Q&A, and then I've also seen some hands raised throughout the presentation. So we can start with one of these questions from the participants, Hilaire. I will now activate your mic. Hi, welcome. Hilaire, I think you were on mute.
I think they may have raised their hand pretty early on in the talk, so yeah.
Okay. Well, we can always come back later.
Yeah.
Let's turn to the Q&A. So the first question, quite interesting. So another source of sound in coral reefs, which is sometimes overlooked, comes from algae photosynthesis, from the oxygen bubbles that
Mhm.
are formed and released, that make a ping sound. So this photosynthesis is hypothesized to be a sonic landmark of coral reefs under stress.
Mhm.
And how much of this sonic contribution was addressed in the labeling of soundscapes in the data set that you mentioned at the beginning of your talk?
Yeah, I'll say that I'm aware, yeah, of the at least one or two studies that have come out and like really cool, described sounds coming from photosynthesis. I would say it has not been, we didn't look for target that for labeling during our efforts. We're very focused on the fish sounds, and there are other biologically related signatures on the reef. So that's not something that we've looked at. I do think it's really interesting to think about these sort of unexpected sources of sound and whether or not they are markers of other, of stress or of something else going on the reef. But no, like we haven't looked at it.
Mhm. That's the, I think, Freeman and Freeman study, if anyone's interested in checking that out, about the daytime sounds produced by algae on coral reefs. How about, yeah, are these reef acoustic playback systems available for purchase, or for what's their production, and do you have plans to share them or make them available?
So they're not available yet. So we are, we've been kind of ramping up our production capabilities and we've continued to iterate on the boards and things. So where, this study, we basically made do with like one really well-working system. And now we have like a couple of dozen. I'd say if you're interested, if you have a project that you think could really make use of this technology, reach out to me or reach out to Aaron. And we can see about, we're starting to try to be better at putting these in the hands of other people, but right now it's still a little bit there's still, finicky things when you're developing technology. And so it's not necessarily super clean that we can just hand it to a user yet. But we are really hoping to get there. And by making, connections is how we start doing that.
Excellent. Um, I think a lot of people will be eager to put this technology into use when it's ready.
Yeah.
Ashik.
Hey there. Hey, great presentation. Really cool study. I was also kind of really intrigued by your background, both from, the science that you're doing and also the policy background and interest that you have. And that's kind of that intersection is my question, which is, I'm in bioacoustics, so definitely a methodology is interesting, scalable, and all of that. But when I look at a kind of a detection and response for a degrading ecosystem like a coral reef, how effective is bioacoustics in kind of creating a more of a faster response, detection and response system? So that's one. And the
Mhm.
second part is that, potentially a visual or a coastal community-based monitoring that could create one local ownership, and second, a more rapid response to some degradation or changes. Are you integrating those data layers to acoustics, or is that a separate process?
So yeah, I mean, you're touching on a lot of really, I think, important points for us to be thinking about as we, kind of structure our research here. One way that I think that our lab is sort of approaching it is we are trying to really work interdisciplinarily with other modes of monitoring and detection on reefs. So like in the Virgin Islands, at this reef site, we have like 5 or 6 PI scientists at Woods Hole, and that includes like a physical oceanographer, a microbial ecologist, chemical oceanographers, who are all kind of putting out sensors and have been monitoring these same sites for a long time. And so we're really trying to start to integrate some of those sort of multi forms of sensing and monitoring, because I do think bioacoustics is not, it's not really mature enough right now on the reef for us to completely use it as a like early detection system, partially because we don't really have enough sort of ground truthing and enough knowledge of what are the acoustic indicators of change on the reef, like, and what are the maybe sentinel species that like disappear first when you start to lose structure or lose some of that like ecosystem function. So I think we're sort of slowly getting there. It doesn't necessarily feel like these systems are completely mature enough to do that yet. But I do think that acoustics offers a lot there. It offers benefit over other sort of visual survey methods and things, in that you can monitor continuously over day and night. You can do it remotely. We're also working on sort of more real-time systems. So like drifters that you could you can put out to drift and or more and listen for sounds of interest, like have a neural net or detector on it and then have it transmit that satellite information back in real time. So rather than having to put out your long-term recorders and then, have them out for however many months, then pick them up, then analyze the data, and then know, oh, 6 months ago, there was, like a pod of humpbacks passing by here, but we like didn't know that at the time. I think moving towards real-time detection and moving towards these like early monitoring response is I think that's the goal of a lot of people in this community. I think the problem is we just need a lot of like legwork to get there. Hope that answered some of your.
Thank you. No, thank you. Great.
Excellent. So we have maybe a handful more questions. And I just wanted to say before that, on the 23rd of October, on the 23rd of April, we will have Francisco, he is, I think, just finished up his PhD at Purdue University Center for Global Soundscapes, and he made a really cool R package for analyzing insect sounds. So put that on your calendars. We'll send an invite for that shortly. But turning back to coral reef soundscapes, I was wondering, in the comparison of, okay, we've detected fish pulses and we're using that as a metric versus measuring the low frequency sound pressure levels. You showed how they were similar in some ways, different in others. Did you compare those metrics with measures of coral reef structure or condition or fish and invert biodiversity? And if so, which one was more tightly related?
So I forget exact. So I believe if you go, there's a paper by Sierra Jarrell, who's a research technician in our lab, and so she went through and took a bunch of this labeled these labeled fish call data and kind of compared it to other metrics, other acoustic metrics that are, like acoustic indices like ACI and acoustic entropy and things. And then I believe was also matching that up with so we regularly take surveys of these sites, including that measure the fish abundance, diversity, and then just benthic surveys of the coral cover and other things. We don't really have a lot of like rugosity data or some of those other measures of the reef structure and things like that. So I don't know how many of those other metrics we've tried to correlate. I know that some of the earliest papers that that our lab put out on the Virgin Islands work did compare, it compared both like the fish abundance and the coral cover, with sound pressure levels, and also found that those were generally, positive relationships. I'm not sure right now about like where we are at with now that we have some more fine scale fish calling rate data, how much of that we've been correlating with all these different things. One aspect that we are really working closely with right now is we have this really cool collaboration with a robotics group at Woods Hole who are developing a robot to kind of intelligently search the reef and like use detections of acoustic activity to guide the robot towards what are potentially like hotspots of activity on the reef. And so that kind of gets at that, like, all right, what are the actual acoustic indicators that might be correlated with activity? And I know that work has been, it's been challenging because picking, all right, is it like snapping shrimp snaps? Is it like fish calls based on our detector that is still imperfect? Is there some other acoustic metric that does really correlate with what you're going to find when you go look there on the reef? A lot of that is still in progress, I'd say. I don't have a particular metric that I think correlated more with the sounds than others right now.
Got it. Okay, let's keep looking at these questions. We've seen, this one was asked a few times, that are there any negative effects of this sort of playback approach?
I mean, I think that's a question that we don't know yet, and therefore that's something that we need to think about and do some work to investigate before we, start trying to hand this out to people wide scale. I think you can obviously think about when I mentioned in my last slide that, the community level effects here are going to be really important because when you put, an added sound source on a reef, it's not just going to attract your one target coral or, fish that you're thinking of. There's other species that are going to be sensitive to that sound and may come in. And so you may actually, be undermining some of your own goals if by attracting corals, you're also attracting the butterfly fish that eat all the larvae, right? So that is that I think we don't know exactly. I can't really think of, extremely harmful impacts that you're going to have if what you're adding is, sound that is meant to basically just replicate what's there. But I can think of ways that it, could alter some community dynamics on small sort of spatial scales, and that may, that then may have cascading effects. So I think that's definitely something we're thinking about and planning by doing, kind of longer term tests of these speakers where we put them out for, weeks or months at a time and sort of monitor the community how it changes over the course of that time will get us, closer to those answers.
Excellent. Yes, I see Sebastian Boll has his hand raised. And why don't we make this the last question since we're at the end of the hour? But what we can do, Nadège, if you're open to it, we have maybe 5 other questions that I can share with you. And if you have some thoughts that come to mind that you're willing to share, we can put that on our YouTube page.
Yeah.
So there's questions here about the parameters of the speaker. If you're interested and open to working in the wonderful place, the Galapagos, with the amazing Gianna Haro potentially. And yeah, a few other questions. So I can share those with you.
Sure.
But right now we have, can you raise your hand again and I can add you to?
I think it was Sebastian.
Okay,
Yeah.
thank you. There you are. Okay, thanks, Sebastian.
Hi, thanks for this amazing talk. I really like the topic, and I think you presented really well. I'm very new to bioacoustics, so so I feel honored that I get the last question. So the idea is that by playing these sounds, the larvae, do you get the sense of there must be like this must be a positive environment for us, so we can adhere or bind, and is that the idea? And then the question is, if that's the case, how would that work if they do bind and then later it seems that it was a false positive signal because there weren't actually fish around. So it is not really a very positive environment for them to grow up in. What's your thought on that?
Yeah, right. There's this like, are we, tricking animals by trying to lead them somewhere where they're not actually going to thrive if the signals that indicate it's a healthy ecosystem are artificial. Obviously that's, not what we want to do. So I think it really comes down to being thoughtful about, okay, what's in a worthwhile application of this. I think a lot about current like nursery settings and reef sort of, outplanting strategies where there's a lot of work being done currently where, corals are attempted to be, spawned either in labs or gametes are being collected from wild spawning corals and then reared in the lab. And in that case, they're often, settled in the lab on small substrates and then grown out for a while and then tried to, outplanted back at target restoration sites. And in that case, techniques to be able to settle more corals at higher rates on more substrates, that can help scale up some of those processes, especially if it's possible that acoustics can supplement for maybe, spending less time conditioning surfaces and prepping them for the corals. So that's, an application of it. And another is thinking about, like just being careful in your site selection. So something that we have noticed, like while we're working very closely with hydrodynamic modeling folks who are looking at, okay, where are places on some of these islands we're working where larvae and particles are likely getting, entrained, but we're not seeing recruitment there. And is there, a way that we could maybe encourage recruitment at those spots? But we have to think about like, are those going to be places that are impacted by, by nutrient runoff or like in the next year that they'll be hit by a heat wave again? There's a lot of other considerations about, the place of where are you trying to bring settlement, and how can you, after they settle, how can you ensure that they survive for longer? So some of that might have to do with the structures maybe that you put in place. There's lots of kind of strategies and work on like developing ideal settlement structures, and sometimes some that are like protected, so sort of layers of tiles or other things on top of each other that might prevent predation like immediately on your settlers once they land. So yeah, I mean, I think that's a really good point, and it's definitely something that like we need to be keeping very conscious at front of mind, that like I said, changing the sound environment doesn't actually change the rest of the environment. So if it's not hospitable for coral growth, it won't be hospitable to the extra settlers that you try to attract there. But if in the process of attracting the corals, you also attract settlement stage fish and they settle there too, then that, and they grow up there and then begin, like it might be a way to sort of kickstart a positive feedback cycle is the ideal. It might probably doesn't work as simply as that in nature.
Okay, well, with that, we'll have other questions answered and more information in the Q&A on our YouTube. And Nadège, I feel like you could do 3 days talking about this method. And yeah, no matter where you go, I hope that you're still involved with thinking about and testing this approach, because it seems like you're really been core to its innovation to date. And yeah, just thanks so much again for sharing your knowledge and experiences with us here. And thanks everyone for joining all around the world. We'll be meeting again in 2 weeks. BioacousTalks is officially back, and it's great getting together, and we'll see everyone soon. So take care.
Thanks so much for having me.
All right, bye-bye.